Welcome everyone. By this video today I am going to give you a brief way on by which you can cultivate a microalgae in a raceway pond system. For that, initially you have to understand what is a raceway pond. It is a shallow pond have a open channel cultivation system designed for growth of a microalgae in a large volume of water. So pond has usually oval loop with the paddle wheel that continuously circulate the culture medium. And what does that circulation ensures? This circulation ensures a uniform exposure of sunlight, proper mixing of nutrients, prevention of sedimentation, and exchange of gases with the atmosphere. Uh, Beside that, on the raceway system, there is other stuff like inlet, outlet, culture medium, and algal biomass. The principle of microalgae cultivation on a raceway system, basically, we have to consider uh, it is a general process which it's happen through the photosynthesis on using natural sunlight, carbon dioxide, water, and nutrients. Algae produces such a biomass rich in proteins, lipids, and carbohydrate through the photosynthesis. So, in raceway pond system, the environmental conditions are maintained to maximize algal biomass and productivity. For the cultivation of algae on a raceway system, normally in laboratory scale raceway pond cultivation, we need a raceway tank or mini raceway setup, paddle wheel for circulation pump, as well as microalgae inoculum is our preference, and growth medium to grow them, and the carbon dioxide supply uh, and pH thermometer and light source analyzer, and also aeration system. Beside that, we need analytical tool like microscope, spectrophotometer. The laboratory procedure for cultivation of a microalgae in a raceway pond include preparation of algal growth medium initially according to the strain that we have selected, and we take the nutrient ingredients like sodium nitrate, potassium sulfate, magnesium, potassium, yes, phosphate, and magnesium sulfate, calcium chloride, and trace other elements. For the setting of the raceway pond system, we usually maintain a shallow depth between like 15 to 30 centimeter to ensure maximum light penetration. As inoculum, we add actively growing microalgae in the pond under aseptic condition. So it is recommended for circulation and mixing. We start the cultivation with the paddle wheel to circulate the culture continuously, and proper mixing, which ensures the settling and improve the nutrient distribution. For light and temperature control on cultivation systems, they need sufficient light for photosynthesis. So, in laboratory scale conditions, we can utilize the light that emitted from sun, or the artificial light we can supply through the electricity. Normally, the light density required is 2000 10,000 lux, and the temperature normally between 20 to 30 degrees centigrade. But in outdoor system, it is preferred to use the sunlight as a natural source of light. For carbon dioxide supply, we supply through the bubbling, and that ensures the algal growth and maintain the pH of microalgae as well while growing on their growth conditions. But the recommended concentration of carbon dioxide is 1 to 5%. For monitoring growth, usually we look up on like optical density through the spectrophotometer for cell counting. Normally, we use microscopes, and harvesting can be done with the different techniques when the sufficient biomass is produced. Like we go with the filtration, centrifugation, sedimentation, and flotation method. The important parameter that we have to consider includes the microalgae cultivation uh, that depends upon the pH, temperature, light, mixing, nutrient, and carbon dioxide. So why we prefer to use a Raceway Pond system instead of photobioreactor? The reason behind is they are economical for cultivation of microalgae and easy to operate in compared to other automated system and mechanical systems suitable for large scale production as well, and they are more energy efficient. And also in terms of limitations, they can easily maintain, or we cannot do maintenance of it. We can utilize the natural sources of light and lower the operational cost compared to bioreactor system. Thank you so much, everyone, for your attentions. I'll be back with another new video with the new content. Hope to have understand the all the matters that I have discussed here. Mm, so thank you so much.